All right, so we're a couple of minutes past 11. I think if people are going to be joining now, we can just sort of join in as we get going. So I don't want to keep everybody too long. But yeah, so all right. Hello, everyone, and thank you for joining the webinar today. My name is Dylan Brether, and I'm a scientific sales executive at Rapid Novor. Today, we're going to be discussing current antibody validation techniques, their shortcomings, and some possible solutions. So I wanted to start today actually with a quick poll, and I'd like to know how many of you have actually had a new antibody or a new antibody lot not perform as expected. So I know myself personally, when I was completing my master's, I was lucky enough to have the opportunity to perform a countless number of Western blots, as master students do. I recall one instance actually in which I was working with a knockout cell line that looked like it was still expressing the protein. So after some mild panic that I ruined my entire experiment, with some additional work, I was able to determine the antibody was actually cross-reacting with another protein within the same family. So with that, it looks like we've got most people have answered so far. So I'm just going to end the bullet and share the results with everyone. So as you can see, it looks like just under half of you have had this happen to you. For those that have not had an antibody perform unexpected, very jealous of you. That's quite amazing. But for the rest of you, you're not alone. So to highlight this problem, I wanted to share a couple of stories with you that might hold a little bit more weight than my story as a master's student. So first, I want to introduce you to Dr. Peter McPherson. Dr. Peter McPherson's lab at McGill was working on a project focused around the ALS disease gene C9ORF72, or chromosome 9 open reading frame 72, and its corresponding protein. So throughout the project, they tested 16 different commercial antibodies directed towards the protein and found that only 2 of those were able to recognize the protein in the Western blot. And only one of those antibodies was actually able to successfully capture the protein via immunoprecipitation and recognize it specifically with immunofluorescence. So this is a great example of not only how improper validation can affect researchers, but also highlights the efforts that individual labs are putting in to ensure that antibodies are properly validated now and in their work moving forward. So another example I wanted to bring up is the example of Dr. Nedenhofer's group. We're testing the prognostic value of a biomarker for personalized cancer therapy. And they were using the highly cited monoclonal antibody 8F1 to detect levels of ERCC1. Now, however, during the course of their work, they actually found that the antibody was not binding specifically to ERCC1, but also was cross-reacting with another protein, CCT alpha. So this cross-reactivity actually made 8F1 antibody unable to discriminate between normal and malignant cells. And ultimately, these findings actually resulted in 8F1 no longer being used in a clinical setting. And unfortunately, the cross-reactivity of 8F1 is not at all a unique example within the industry. So each of these 5 antibodies are sold as being specific to CDK1. However, as demonstrated in the blots, you can see that the 2 antibodies on the left are also cross-reacting with CEP152, while the 3 antibodies on the right don't show such cross-reactivity. Now, it may seem that there's a simple solution to avoid this, and that would be to only use the antibodies on the right when performing your research. But it's not quite so simple. And that's because one of the main issues is that the best resource that we have as scientists and researchers in choosing antibodies is basing that off of previous publications. And in this case, the cross-reactive antibodies on the left were actually been cited in over 200 papers. So to take a quote from Dr. McPherson, the problem is that many, perhaps more than half of commercially available antibodies don't target the protein the manufacturers claim they do, or at the very least, they may recognize the intended target, but are also cross-reacting with non-intended targets as well. And as it turns out, Dr. McPherson's estimation was actually quite accurate. In a 2015 study, it was found that 75% of commercially available antibodies were non-specific. In addition, 50% of the 5,000 antibodies investigated were not actually able to be used in their intended application. And on top of that, 56% of the published antibodies could not reliably be traced. And this actually may help explain the findings of Dr. Begley's group. When they were trying to reproduce the primary findings of 53 different publications, they were actually only able to reliably replicate 6 of those results, meaning that 47 out of the 53 publications were not actually reproducible. Now, in addition to these research antibodies, antibodies are also used to identify clinical biomarkers, as noted with the example of 8F1, and they also may display cross-reactivity. So this chart provides a number of examples where antibodies were being used in clinical settings, only to later be found as being cross-reactive. And as you can see, that includes our, our 8F1 antibody, as well as a number of other antibodies to additional targets. So these findings highlight the prevalence of cross-reactive and nonspecific antibodies. And the result of this is an increase in the time, the cost, and the resources spent on research. And it also leads to a decrease in clinical confidence and prognostic value of biomarkers. So at this point, it may feel like I've painted a relatively bleak picture of the current state of affairs, but the good news is that we're actually making great strides in our effort to use and produce high quality antibodies. So the reliability of antibodies is continuing to increase over time. Much of this began in the 1970s with the development of hybridoma technology. And this continued to improve further into the 80s with the introduction of phage display and recombinant antibody technologies. So since then, the improvements in reliability have been less drastic, but that doesn't mean that major steps aren't still being taken. So in the late 2000s, ORCID created an antibody registry. And in 2016, the International Working Group on Antibody Validation proposed the 5 pillars of validation to try and begin standardizing validation efforts. So these 5 pillars are genetic validation, orthogonal validation, independent antibody validation, recombinant expression validation, and capture MS validation. And then finally, the last little note on our timeline here is ABCD, and that's antibodies chemically defined. This is a group that has actually created a database for the sequences of antibodies. So being aware of the current state of antibody validation in the industry, we wanted to hear how this is impacting our clients firsthand and performed a survey to find out. So we found that 75% of respondents experience unexpected results when working with antibodies. And of those, 88% of them actually were not able to identify the cause of these inconsistent results. And surprisingly, that is also with 75% of respondents going beyond just using simple binding assays. So when we also wanted to look at how we can improve antibody validation. And when we did this in the survey, we found 3 key points. Number 1, antibody validation needed to be more effective. Number 2, antibody validation needed to be more scalable and high throughput. And 3, antibody validation would need to be less costly. So looking at the first point, in order to increase the effectiveness of validation efforts, we really need to understand what the current efforts are entailing. And there's a lot of great efforts currently underway with groups all over the world making really big strides and working really hard to improve antibody validation. So in addition to the user-based validation, these include citation-based validation and platforms awarding validation initiatives by manufacturers. And speaking of manufacturers, there are many great antibody manufacturers that are currently working tirelessly to ensure proper validation of their products. And manufacturers are also beginning to include more performance-specific information on their data sheets. This includes positive and negative controls, ELISA titers, and a lot of other extra data, which you can see in this chart. So on top of these efforts that are underway, recombinant antibodies are also becoming more and more regularly used as manufacturers are also moving to recombinant systems. So though more of an effort is being made to properly validate antibodies, the current validation strategies really haven't changed much since 2016. So if we revisit these 5 pillars of antibody validation, each of them is great at confirming the specificity of an antibody, but we still continue to experience issues with regards to reproducibility. So let's do a quick review. So the 5 pillars are becoming more widely accepted, and orthogonal methods for proper antibody validation are becoming the standard across the industry. Number 2, manufacturers are providing extensive resources and additional data for antibodies, allowing proper validation to be done more easily. Number 3, we now even have online databases and resources, along with AI to help us with antibody selection. So what is it that we're missing? Why are we still having issues with reproducibility? Well, what's missing is the sequence. Currently, antibodies are characterized and validated based on what they bind and not their actual physical properties. Now, my question is, would you work with any genes or vectors or oligonucleotides without knowing their sequence? I'm sure that the resounding answer to that would probably be no. So why should antibodies be any different in this regard? And if you're wondering why the amino acid sequence matters, well, the amino acid sequence of an antibody is going to be what determines how that protein is going to fold and ultimately how it's going to function. So changes to the sequence can have an impact on various characteristics of how the antibody behaves. And that includes it can affecting the binding and the affinity of the antibody. So it may not bind as expected if this were to be changed. It also will determine how specific the antibody is and whether or not there's any cross-reaction with other proteins. Now, this, of course, if there's any cross-reactivity, can alter how reproducible results are. And it can also affect other physicochemical properties such as half-life and aggregation. So what might antibody validation look like with sequence confirmation included? So using an orthogonal validation approach, one of the 5 pillars, specificity can be confirmed using a standard method such as Western blotting or via ELISA. And then the reproducibility can be confirmed in orthogonally with the sequence validation confirmation step. So having the ability to perform this type of confirmation quickly and effectively would reduce the total amount of time spent validating new antibodies and lots and can decrease the need for troubleshooting in the future. So though confirming the sequence would be beneficial for researchers and manufacturers in many areas, I want to specifically highlight the impact that such a reduction in troubleshooting time may have for a company that's focused on developing antibody therapeutics. So I'm going to ask you all to use your imagination a little bit here and just imagine a situation that early and often routine biophysical characterization and sequence confirmation was able to make a really tiny impact in the early discovery and development stages of such a company. So first, let's make an assumption that by adding this confirmation step into workflows, there's going to be a 0.05% increase in the probability that a therapeutic candidate will be successful. And corresponding with that, let's say that there is a decrease in troubleshooting time by just 1 month on a 13-year cycle. So using an adapted version of the capitalized cost calculator, what does this all mean? So this could result in capitalized savings of approximately $12.3 million over that span. So after hearing that, you're probably wondering, why isn't this already being done? What's holding everyone back from regularly adding this into their workflows? So one of the main issues is that many groups don't have access to individuals with the necessary expertise and or the equipment. Speaking of the equipment, this type of work requires a mass spectrometer, which is not only costly to purchase, but can be quite costly to run as well. Now, say if you do have the expertise and the funds, the process has historically been relatively slow and low throughput. So this is where MatchMap, which is our new rapid peptide mapping service, comes in. So MatchMap is a routine, fast, and easy solution to verify your antibodies, reduce risk, and increase reproducibility every step of the way in your research. So our technology analyzes and confirms the sequence of your antibody and will match that with the expected sequence and can then also identify any anomalies for future study. And what sets MatchMap apart from previous efforts like this is that it overcomes many of the shortfalls in those previous sequence confirmation protocols. So MatchMap provides fast results, confirming your sequence in only a couple of days, as opposed to possibly up to a couple of weeks. And it's cost effective, which will allow it to easily be integrated into your workflows. We've also created subscription plans, which combined with our ability to process hundreds of samples a week, you can quickly and accurately confirm the sequence for every batch of the antibody you need, which will definitely bolster up your quality control efforts. And then finally, it can also be used to confirm the sequence of your antibodies for any sort of patent applications. So the workflow itself is actually quite straightforward in terms of what we require and how it's done. So as we receive the samples, they're digested and then processed by our team. They're then run through one of our mass specs to undergo real-time peptide mapping. And finally, the sequence is then confirmed and double-checked by our bioinformatics team. So that's great. What do we need to get started? So all we need from you is 20 micrograms of your monoclonal antibody of interest, as well as the expected protein sequence as a reference. So we can work with any species, any isotype, but if you do ever have any concerns, our team is here to answer any questions and have done a number of these projects and have more expertise than anyone else out there. Now, what if you don't actually know the sequence of your antibody? So there are still a number of ways that we may be able to help out. For starters, our team can actually help derive the protein sequence directly from your protein sample using either our remab monoclonal sequencing or repab polyclonal sequencing technology. Alternatively, if it's a relatively common antibody, some antibodies actually have their sequences published online, such as in the Antibodies chemically defined database. So it may be worth checking these to see if the sequence is already known. And if you need a hand with finding out which databases to look in, feel free to reach out as well. Or if you have access to the original hybridoma or cell lines, NGS can be your friend and can help get you the sequence that way. And then finally, if it's a commercial antibody, you can actually ask the manufacturer if they'll work with us to MatchMap and confirm that antibody and the new lot for you. So with regards to that last point, how might that work? So put together a quick little diagram. So if you were to ask your manufacturer to MatchMap it, they can send the antibody sample and the expected sequence to us. We could then confirm the sequence, send the data back to the manufacturer, who then in turn can provide you with a MatchMap confirmed antibody and peace of mind knowing that you can expect reproducible results now and for all your future experiments. So At the end of the day, our goal really is to increase access to the necessary tools for both researchers and manufacturers to ensure that antibody validation continues moving forward. For manufacturers, our remab and repab technologies can allow them to move towards recombinant antibodies, which have higher reproducibility. And as well, we've been partnering with both service providers and antibody manufacturers to just improve antibody validation efforts in general. So as a quick conclusion, what we focused on today was MatchMap, which we're offering as a low-cost, high-throughput addition to routinely check the sequence and properties of the antibodies you're either using or developing. So we're offering this with subscriptions and also some free trials to any qualified clients. So if you'd like to talk about how this fits into your routine or can add on to your quality control practices, please feel free to contact me. So with that, it's a relatively short and sweet presentation, but if there's any questions, feel free to put those in the Q&A section and we can see if we can answer those for you. So one question we do have, do you know where this recording can be found so I can refer back to it? We will actually be, this has been recorded, we'll be posting it to our YouTube channel as well as to our website. And we can also, I believe we'll also be sending out a recording of it once we actually have finished it and just cleaned everything up. And just see if any other questions come in. All right, doesn't look like we have any other questions at this time, but if you do have any questions, feel free to reach out to me directly. I'll send a quick follow-up with the recording as mentioned before. But yeah, thanks again for joining me, and yeah, it was great to have everyone. Oh, it looks like we do actually have another one in terms of the cost involved. So right now we are offering, as mentioned, we do have free trials that we're offering to clients that we can actually qualify and work with. In terms of the standard pricing, if we're doing a one-off conformational sequence, right now our price for that is $1,000 per antibody. But we do offer, as mentioned, subscription plans, so we can also look into that on a more individualized basis as well, depending on your needs.